The beauty and the complexity of cell and gene therapy is that every patient, every donor, every manufacturing process, and every final product is slightly different. And so there is no one-size-fits-all answer for cell and gene therapy.
That was Priya Baraniak, Chief Commercial and Development Officer and board member at Pluristyx, a leading innovator in induced pluripotent stem cells, helping accelerate the development and commercialization of revolutionary cellular therapies. With proprietary mRNA reprogramming, immune cloaking, and safety switch platforms, Pluristyx empowers cell and gene therapy developers with faster, safer, and more scalable starting materials. Pluristyx is fueling breakthroughs across regenerative medicine, immunotherapy, drug discovery, and emerging organoid-based technology. Welcome to the Biotech Stack, where we explore the cutting-edge technology fueling the future of life sciences and biotech. From next-generation lab automation and AI-driven drug discovery to breakthrough biomanufacturing and research tools. We dive deep into the innovations transforming health, food, energy, and the businesses behind them. I'm Corinne Regan, your host. In today's episode, I'm joined by Dr. Priya Baraniak, Chief Commercial and Development Officer. We chat about her journey from academic biomedical engineer to a startup builder, commercial leader, and now a driving force behind Pluristyx's mission. We chat about Pluristyx's innovative technologies and their aim to simplify and de-risk the complex process of cell therapy development. Let's get started. Well, Priya, thank you so much for joining us today. I'd love to kick things off, and I'd love it if you could walk us through your background and how you got into this space.
Thanks, Corinne. It's an honor to be here, so thanks for the invite. I am a biomedical engineer by training. So my undergraduate degrees were in biomedical and electrical engineering, and then I did a Ph.D. in biomedical engineering and postdoc as well. I always thought I would stay in academia and had a grand plan for my own research lab. And at the 11th hour, jumped ship to industry, where I landed at a small clinical stage tissue engineering startup, really, that was working on stem cell-based tissue and ligament regeneration constructs. And so right up my alley as a stem cell scientist and tissue engineer at that point in time. And I was predominantly on the science side. I was hired as a scientist and then quickly took over running the scientific programs there. But because it was such a small company, I really had the opportunity to interface with manufacturing, with quality, with regulatory, to participate in writing a 510(k) filing and speaking with regulators, and even writing claims for patents and prosecuting them with the USPTO. And so really just an awakening to industry and everything that it takes to move a product from concept and R&D to the clinic, and a real education in the 13 months I was there. And that quickly came to an end, a very unanticipated end, when the board of directors overnight, that was predominantly VC funded, decided to halt all operations and instead fight a legal battle around IP. And so I found myself wondering what to do next. And I was incredibly lucky, and it was very serendipitous to have John Rowley, who's the co-founder of Rooster Bio, reach out and say, "Hey, I'm starting a company. I need someone with your background and expertise. Will you join me?" And the value proposition was really clear to me. It made perfect sense. And so I said, sure, and took the leap. That, however, was more of a commercial role in helping to build a commercial engine at Rooster, sales, marketing, business development, all of that, which I knew absolutely nothing about as a trained scientist. And I told John as much. But again, I was really lucky that he was like, "Yeah, you'll figure it out." And he gave me that opportunity, and we did figure it out. So I was there for the first 5.5 years with Rooster, and then was recruited to go help start another company down in Miami, which was also a provider of cellular starting materials. And so there built a birth tissue and an apheresis business, and got to wear multiple hats again. So not only on the commercial side as chief business officer, but I also got to head process development and manufacturing and serve as laboratory director there. So really got to immerse myself in the science as well as the commercial. That was a step away from me from what I was used to doing, which was always around stem cells and tissue engineering, regenerative medicine, cell therapies, into more immunotherapy, immuno-oncology, and the whole realm of, CAR Ts and those sort of therapies, which was really, really cool. So it really broadened my knowledge base and my horizon. of the cell and gene therapy space, which was very value adding for me personally and professionally. And then about a year ago, I joined the board of directors of Pluristyx. And then in April, towards the end of April, officially took the chief commercial and development officer role. So I have a board member role as well as the commercial role here. I've been friends with the co-founder and CEO of Pluristyx, Ben Fryer, for over a decade. And so have always been a champion of the company, followed the company and the tremendous progress they've made, and also know Ben to just be a fabulous human being. So it's been wonderful to join this team and now work alongside them.
Very cool. What a nice journey. I'd love to focus a little bit more than what on Pluristyx. What does Pluristyx do? And then a second question, we talk a lot about commercial. Can you dive a little bit deeper into when you say commercial, what does that mean? And what does a commercial strategy look like in a company who's selling a service, tools, or technology?
Yeah. So Pluristyx is a provider of induced pluripotent stem cells and gene editing and gene engineering technologies to cell and gene therapy developers. So again, we are a starting materials provider to therapeutics developers. What the company has is proprietary mRNA reprogramming technology. So it's footprint free and it circumvents a lot of the limitations with, say, the use of Sendai virus for reprogramming. Our methodology also is a lot quicker. It's 28 days for reprogramming of cells versus 60 or 90 days for other methods. So you get a younger, healthier population of cells when you're done reprogramming them. And we also have a process that results in a polyclonal population, which again is very unique. Instead of clonal expansion, where you pick a clone early and you're serially cloning that clone, we have a polyclonal population that is much more akin to an actual embryonic stem cell, which is polyclonal in nature. The cells are younger, and we bulk edit, bulk modify the cells. So in a single line, you can actually screen thousands of clones and not have to select a clone until downstream, where you know that that clone is not going to have any sort of genomic drift or instability, and will also functionally meet your requirements for your therapeutics program. So significantly de-risks your program as well. We have, as I mentioned, proprietary gene editing and engineering technologies. One is an IAC stealth immune cloaking technology that helps the cells evade the immune system. So in the allogeneic transplant realm, you circumvent a lot of the issues that you might otherwise have if you cannot perfectly HLA match the donors around rejection or an immune response to the cells. But if you are evading the immune system with iPSCs, you could be causing another safety issue, which is teratoma formation with any cells that are left undifferentiated and proliferating unchecked. And so what we have is the only commercially available safety switch called failsafe. That is a kill switch that any differentiated cell will, continue to persist. And with the kill switch, when you turn it on, any undifferentiated iPSCs are eliminated. So you don't have that safety issue by evading the immune system. And so this becomes the basis for any, iPSC-based cell therapy or gene therapy that is being developed. These cells are also, iPSCs are also the basis for a rapidly growing drug discovery, drug development market. And especially with FDA 2.0, the Modernization Act, and now the push globally, we just saw something come out of the UK late last month mandating significant reduction of animal testing and moving away from animal testing as a whole by 2030. We're tracking to that here in the United States as well. iPSCs are really the basis for these organoids or new approach methodologies, organ on a chip, and microphysiological systems to really bring them to an industrial scale where they're standardized, robust, and reproducible, and can be a surrogate or an even better predictor of human response to drugs and cell-based therapies.
Gotcha. Now, from the cellular material standpoint, are you providing the cellular material and all of the things you just mentioned? Or are you also doing some of these services in-house where if somebody has some type of drug of interest or a pathway of interest, that you guys can actually work on your own cellular material as well?
Yeah, we have both. So we're not a therapeutics developer. That is not part of our mandate, if you will, or our mission. We're here to enable thousands of therapeutics developers and drug developers. But yes, if someone were to come to us, say, with their own donor material for autologous use, or, hey, we've identified an allogeneic donor, like we've been using apheresis material from this super donor, but we know that at some point this donor is not going to be recallable. So we'd like to generate a bank of iPSCs from this donor, and we have the consents to do so. We can reprogram that donor's cells. So now you have an evergreen source of your super donor that you know works for you. Or again, in an autologous setting, if there is a patient who needs their cells reprogrammed for autologous therapeutic use or for drug screening in a very personalized way, we can reprogram those cells. We can also engineer and edit the IACT and failsafe, or do custom gene editing and engineering for anyone into any cell line, whether it's an iPSC or another cell type that they want engineered or edited. We could do that for them as well.
I see. So then going back to this commercialization component, this strategy, when you talk about like small molecules or biologics, even they've been on the market for so long, and the understanding of how technology works and how you go from A all the way to clinical seems like a pretty clear path.
Right.
You mentioned a lot of moving parts, even just within this product offering you have. So what is that strategy? How do you present this offering in this solution to the market?
Yeah, a lot of it is still education, right? And going out and really understanding the uniqueness of each program. The beauty and the complexity of cell and gene therapy is that every patient, every donor, every manufacturing process, and every final product is slightly different. And so there's no one size fits all answer for cell and gene therapy. When you're dealing with a molecule, something that is, chemically similar time and time and time again and very defined, it's a lot easier to set these critical process parameters, critical quality attributes, and say, like you're saying, if I do this, I will always get this outcome. With living biological systems, there's just so much variability. And while we control so much, Mother Nature really controls the vast number of things, right, at the end of the day. And so we have to always account for that. And so it's not the commercial process or the sales and partnership process is highly consultative, right, where you really have to sit with your partners, with your customers, understand their specific programmatic needs, their endpoints, and tailor a solution that meets those needs, but within that can be robust, reproducible, and economical, right? So that's what we work to do with customers and partners. And again, a large part of it is education, because a lot of times we don't know what we don't know, and we don't even realize as an industry that we're in pain until someone says, hey, it doesn't have to be that hard. We can do this differently, and it can be easier, and we have a solution to offer you. So it's, again, it's highly consultative. It's really a mind-sharing exercise and a thought exercise where you want a partner who is not just buy this from me and good luck, but someone who can really sit with you, understand those pain points, and tailor a solution to you. The other piece that iPSCs, where they come in and they offer so much tremendous potential is around standardizing a lot of this and having a reference standard material that you can always go back to, and it's going to have the same quality attributes, at least at the start, versus a primary cell, right? Even from the same donor, every time you draw that material, it's going to be a little bit different based on, did they have a cold? Did they go out and just had a rager the night before? Like, who knows, right? There's any number of things that is going to affect whatever you're getting is truly just a snapshot in time when it's primary tissue or primary blood, whatever it might be, right? Cells from blood, et cetera. And so iPSCs provide this source where you can bank back and always have a reproducible starting material that you can reference back to. And that's one thing that largely holds the industry back still and is a critical pain point, is just reference materials and standardized starting materials that we can rely on.
Now, are there a lot of companies in your space where you are a referenced material or there is some more standardization? Is this part of this workflow growing because of that?
Yes, that's part of what we're working on now. And again, with the push towards NAMs and organoids and moving away from animal testing, there is even more of a need now to really set some reference standards and be able to industrialize what is still really an art and bespoke science where there's so much variability from lab to lab, institution to institution. And that really holds us back, not only in reproducibility and being able to move these programs forward individually, but as an industry, it makes it hard to compare large data sets, really compare apples to apples and say that we can have confidence in the data that's coming out, let alone that it might be predictive of outcomes, right? Which is really where we need to move towards.
Okay. So in addition to what Pluristyx has as a core offering, it looks like you've also done some partnerships along the way. I think it was back in 2023, there was a merge with panCELLa.
Correct.
What exactly influenced that decision, and what does that do for the offerings that Pluristyx has now?
Yeah, so this obviously predates me at Pluristyx, so I can speak to it to an extent, but not in great detail since I was not a part of that deal. But panCELLa had the iAct and failsafe technology that has been licensed now and is entirely owned by Pluristyx. So we licensed in that technology. We acquired panCELLa. That integration is complete. So that technology now belongs to Pluristyx with the right to, freedom to operate, to sublicense that technology to our customers and partners. And we have then iterated on that technology, right, to further refine it and make it even better for industrial use and clinical use.
So then touching a little bit more, you've touched a little bit on some of the customers, but what is that ideal customer for Pluristyx?
Well, I think an ideal customer for anyone is someone who has money. Let's start with that. In cell and gene therapy, you are capitalized and you're going to be around. No, all joking aside, although not really joking, there are so many different flavors of customer, if you will, for us, because there are several market verticals, again, that iPSCs really feed into, right? They feed into the broader regenerative medicine market for tissue engineering and additive biomanufacturing, like bioprinting and things like that. There is the pure cell and gene therapy space, again, right, where cell-based therapies or cell-based gene therapies, where you need iPSCs. There's drug, and then there's drug testing, discovery, toxicology testing that they fit into. So really our customers and partners span the entire stakeholder chain in broader regenerative medicine, if you want to think about it that way, or drug development, whether that, again, it could be small molecule or not large molecule where they need to test these, right? And iPSCs, again, can afford them that standardized source for testing their drug products, and even in a very personalized manner, all the way to our actual drugs that are living cell-based therapies.
Gotcha. And then from a regional standpoint, do you find this to be like a North America focus, or is there a global expansion for you as well?
We do have customers outside of North America, but the vast majority of our business is currently in North America. Now, some of that is just because we're a North American company, we're an American company, and we're a small company. Our entire team is 12, right?
Okay.
And our commercial team is teeny tiny. And so we're just scratching the surface. The amount that this organization has been able to do with a very small team is really remarkable, right? Absolutely small, but tremendously mighty team that we have of brilliant scientists and very passionate and dedicated individuals who are able to carry a very heavy load with a lot of grace and execute flawlessly, which is fantastic. Again, it's so terrific to be a part of this team. And as we move into 2026 and beyond and increase our commercial team size, commercial efforts, definitely, I think we'll see more global expansion. North America is still the biggest market, but obviously there are large markets, especially as you look at some of these, again, directives that are coming out of the UK, Europe around organoids and NAMs. There are larger and larger market segments in Europe. And then, of course, Asia, right? China, huge market opportunity there as well.
Do you know anything about the China market? Are they also pushing for organoids and NAMs to be kind of standardized as well?
I have not seen anything, but I can't say that I would call myself the expert on APAC. So I'm
Yeah.
not sure. It's possible that there might be a push and we've missed it, but I have not seen anything.
So what kind of differentiates Pluristyx from maybe others on the market? I know there are probably others that offer this part of the workflow. What would you
Yeah.
say is your,
Y-
I don't know, special
Yeah.
sauce?
So again, some of it, a lot of it is around the actual science and technology itself, the mRNA reprogramming, as well as the IACT and fail-safe technologies that are all proprietary to Pluristyx and different from what others are doing. In some cases, the differences are very obvious, Sendai virus reprogramming versus mRNA or episomal reprogramming. There are others who do mRNA programming, and then the differences are a little bit more nuanced, obviously, there, right? Clonal versus polyclonal, and some others. Where as we start to delve into that, I'll be honest, I really realized that I'm an engineer and not a biologist as we start to get deeper and deeper into this. And I'm like, oh man, I got to go back to a biology textbook and remember exactly what you guys are talking about as they get into gene editing and all this stuff. But there's some of that. And then with the IACT, the immune cloaking technology, a lot of what's out there is more of a knockout technology. So knocking out certain genes, B2MC2TA, for instance, there's some CD47 technologies out there, like what Sana has that's proprietary to them. And then some HLA knockouts, HLA-A, G, E, that people do. Those, however, do have some limitations where you might still have to immunosuppress the patient because you are still dealing with NK and macrophage activation and response. So unlike those with IACT, we're actually knocking in or adding in 6 factors that cloak the HLAs from the immune system. So it's like a lighthouse in the fog where the lighthouse is still there, but there's just fog covering it. So you don't have that same NK and macrophage issue. So you don't need to also immunosuppress or also do something else to the cells to truly evade the immune system. So that's one key differentiator. And then the other is the fail-safe technology, as I mentioned, we're the only commercially available safety switch on the market today.
Okay. Touching
I'll
a
add
little
one
bit.
more thing. Sorry.
No,
So that's
please
all
do.
that's all the science, but coupled to that and adding to that is the one-stop shop element, right? Since we own all this technology, you only have to license with us or do business with us to have access and the freedom to operate with all of this today. With other technologies, you also have third-party sublicensing where you have to go to a third party for access to that technology that's been incorporated into the cell line you're using. So that encumbers the process because now you have multiple contracts with multiple entities that you have to execute to be able to move your program forward. And on top of it, you have stacking licenses, annual fees, royalties, which also makes the cost balloon. So from both a scientific and a business perspective, and especially when you put those together, that's where we're really differentiated in the market.
Gotcha. One solution. Can you dive into a little more detail on the licensing? What do you mean by there's different licensing agreements as you're going through this process?
So in the iPSC space, it is a transactional sale sometimes when it comes to just using the cells for R&D or preclinical use. But as you move into clinical use of the cells, because the IP landscape is so fraught and very busy, you do have to license the cell lines of the technology that's embedded in the cells, essentially, for you to be able to use them. So what this normally looks like is some sort of upfront payment for the license. There are often annual fees, annual maintenance fees for the license. And then on top of it, usually milestone fees, right? If you get to first in human, as you move through clinical trials to phase 2, 3, and then into commercial, additional milestone payments that might be due. And then there could also be royalties on top of that. So once you're commercial, you may owe royalties in perpetuity or up to some ceiling. There are any number of ways to slice and dice all of these, but typically this is the structure for iPSC use for clinical and commercial use is some sort of licensing agreement. Now, let's say that you're using Sendai or CRISPR-Cas9, right? Now you might be getting your iPSCs from a certain provider who has manufactured those for you and can provide you with the banks. But that technology, that Sendai virus technology or that CRISPR-Cas9 technology that has been used and incorporated actually belongs to someone else. And the entity that has manufactured those banks for you does not have the rights to sub-license that technology to you. So now you have to go back to the Broad Institute or whoever it might be and execute a licensing agreement with them and a cost structure with them on what those payments might look like on top of what you've already or are negotiating with the manufacturer of those lines.
I see. But because you guys have all of these in-house, there is one singular licensing agreement. That seems like a very huge advantage.
Yep. And it's with us. Yep. Exactly. One throat to choke. Not mine.
Yeah. No, that's interesting. Everybody always talks about the one solution. Everybody wants to be one solution, but that's a layer of complexity I wasn't aware of. And it does seem nice to have that as one single point of contact.
Yeah. And again, oftentimes we think as scientists, we're so focused on the science, right? That it's not until we're well into it and we've already kind of pot committed, right? We put all our eggs into this basket. All our preclinical data is with this cell line, manufactured this way under this CMC, only then to look at the business side of it and realize, oh boy, we just set ourselves up for a whole lot of pain and costs that we didn't anticipate. Or, it's just the thread that unravels, right? You start to pull that and all of a sudden you can't get all the licenses or deal structures aren't working out. And you can not only cost yourself years and millions of dollars potentially, or over the course of years, millions and millions of dollars additional because of that, but entire programs could go under because of this sort of thing, right?
I see. Yeah. So when we're going back to the how do we commercialize this, this is part of that educational component, that this part has to be part of the conversation.
Yeah. Yeah. Always start with the end in mind. And that's not just, again, from a scientific and manufacturing perspective, also from a business perspective, right? What do you need for your program to be commercially viable, for your investors, to meet your investment milestones and your investors to continue to invest, or for you to become self-sustaining? And from a business perspective, are you actually on that path or not?
Gotcha. So shifting gears a little bit, we've touched a little bit on organoids. I've seen some of your chats at conferences. This seems to be a big area of focus. Do you mind going into a little more detail on what organoids are, organ on a chip, and why this is such an important move in the industry?
Yeah. So organoids have been around for decades. This is nothing new. And even organ on a chip technology has been around for more than a decade, 2 decades, more than that, right? And so it's not new technology, but it is technology that, again, really has been a bespoke art or more of an academic science and not really completely industrialized. Now with this focus on moving away from animal testing, 90% of preclinical animal models are not predictive of human outcomes. So we, go through all of the cost of testing animals. There's the ethical piece of, sacrificing these animals, subjecting them to sometimes really brutal regimens in the name of science and clinical science. And especially in the case of non-human primates, I think for a lot of people that cuts even closer to home, right? And so there's a real push now globally, or again, at least in Europe and in North America, to move away from animal testing and to move to models that are much more predictive of human clinical outcomes. And that's where organoids, organ on a chip, and microphysiological systems have really come back up. And there are a number of pieces of this. I was just at a conference in Charlotte last week, arranged by Wake Forest, that was solely focused on this, all the various elements from the cellular starting materials to biomaterials to manufacturing processes, in silico models, digital twins. So AI and ML being brought to bear on this, cold chain logistics, cryopreservation, storage, all of these things that must come together, and analytics as well, to really move this forward in a meaningful manner and make it, again, industrialized, standardized, robust, and reproducible. And so organoids are little balls of cells, really, right? So with iPSCs, for instance, we can force aggregation of the cells. They like to be aggregated. They are adherent cells. So they will adhere to one another in culture and form these spheroids, these balls of cells. We have a ready to differentiate product. We call it our RTD iPSCs. It's a and a pluriform kit. It's a very unique offering for the industry. Normally, what you have to do is spend weeks culturing and expanding cells to have enough critical mass to then generate thousands of these spheroids that you can then differentiate into these organoids, which are little mini organ systems, essentially, in these balls of cells. With our high density, ready to differentiate pluriform kit, where the cells have already been suspension adapted, you can thaw the cells directly into suspension culture, and overnight you have thousands of uniform spheroids that are ready to differentiate. So you take weeks of the workflow out of your process by starting with that robust, reproducible material. And again, variability, right? You might have some variability in that expansion process and the end product of the cells that are going into the spheroids here. You're thawing vials that are QC'd and released to a certain standard every single time to start that workflow. So then to create organoids, you take those spheroids, and now you can either introduce different chemical agents to drive differentiation and commitment to different lineages, whether it's neural, maybe you're trying to go after a liver organoid, a kidney organoid, neural for brain, cardiac. Those actually beat, which is the coolest thing. I've been doing this since I was an undergrad. I don't care how many times I see it. When you see them beating, it's like, wow, this is so cool. So you could take it, intestinal organoids. People are doing all kinds of things, right? So you can create these little organ systems, essentially, with these balls of cells, either with chemical induction or, like I said, delivering different factors and causing them to then differentiate to these different cell types, right? So then you can interrogate those. You can either interrogate them with, let's say hitting them with a chemotherapeutic drug or a drug and seeing, hey, do my cardiac cells die off? Or does it cause arrhythmias in them where they're all beating synchronously and now all of a sudden, I've got some issues with the beating when I administer this, or there's a certain threshold upon which it becomes cardiotoxic or toxic to the liver or to the kidneys. You can use those to 3D bioprint complex tissue architectures, right? If you're a tissue engineer and 3D printing, or you could seed them onto a biomaterial scaffold and make maybe a tissue engineered blood vessel where you have several different cell types that are needed, smooth muscle, endothelial cells, et cetera, for an actual functional blood vessel. So the possibilities are limitless, really, for what you could do with them.
And I love the fact that we're moving away from animal models. You mentioned that in Europe and overseas, there is some more movement towards organoids and NAMs and getting away from animal models, but that it's coming in the US. Do you have any insight into what that timeline looks like? Has there been conversations around where's that regulation coming into play? And
yeah, there have been. The FDA has started to release some guidances around this. NIH, NIST, there are a few different agencies that are involved in this. We haven't been given a timeline just yet in the US. I would assume that is coming. Like I said, just last month, the UK actually released a timeline as well as government funds to really push this forward to meet those timelines for phasing out animal studies. And so hopefully we'll see something similar in the US where it's not just, okay, go, but like, where do you want us to go and when do we need to be there? So we can start to track to that. Yeah.
And then speaking a little bit more about this cell and gene market therapies, they're a little bit more variable. There's a lot more nuance to it. What are some of the challenges you see as a whole in this industry, and where are you guys positioned to help to tackle some of these challenges?
Yeah, again, standardization, that was something even last week in every conference I'm at, standardization comes up, right? So as I mentioned, across every unit operation, across the supply chain, across the workflow, we really need to find ways to standardize. And all the way from the starting materials to the manufacturing processes, how we're assaying these and making sure that the data is comparable and that we can use it in a meaningful way. AI, ML are great tools, but again, they're only worth the data that we feed them, right? And the fidelity and how good the data is that goes in, how much data and how good it is. So these are all things that we continue to talk about as an industry. I think I'm definitely optimistic based on the number of partners who are, willing to come together to share data, work together collaboratively to make this happen rather than seeing competitive positioning in the industry. We have to collaborate, right? A rising tide lifts all boats, and no one company has the expertise and all the tools and technologies that are necessary to really execute to these complex therapies and workflows. So collaboration is absolutely critical. And again, I think we're making strides in that. And in a largely capital constrained market as well, you see more and more of these collaborations and creative ways of working together and putting one and one together to make four, right? To move therapies forward, to move the industry forward, and to also, quite frankly, make capital investment more lucrative by using these kinds of collaborations. So recently, we've launched something called the Organoid Commons. It's a consortium. The Commons stands for the Consortium for Organoid Manufacturing Measurement and Optimization, and a network for standards. We have 9 other industry partners who have already signed on as founding members of the Commons. And together, we have a lot of the complementary tools and technologies that are needed to move this organoid science forward and make it more of an engineering discipline where it's reproducible, standardized, set some of these standards, push out standardized workflows and products that the industry can use, and really move this forward in a quick and meaningful way. So we have cellular starting materials, tissue suppliers, we have the reprogramming technology and the iPSC technology. We have providers of media and reagents and biomaterials and matrices, transfection reagents that are needed for these processes and workflows. And we have providers of different, consumables as well as manufacturing platforms that are needed to move this forward. And so we're starting to, in 2026, we'll really start to move towards some very tangible outputs from these collaborations.
That's great. Now, are most of these partners also smaller startups who are innovative in the space? Do you find any big partners that are joining some of the big guys that may have acquired some startups along the way?
Yeah, so some of the ones who are in the public domain, not all of them have been publicized yet, but we do have Biolamina, we do have Solesis. Not a lot of everyone might not be familiar with Solesis, but they are the parent company for Charter Medical, as well as CCAM and Polyzen, which are biomaterials companies. And then we have Discovery Life Sciences. That's a large company as well. Yeah. So we do have some very large partners signed on, and others who, as I said, we haven't publicly disclosed yet, but in coming months will become public knowledge.
You guys have just received a successful investment from a few partners.
Yeah. So what that allows us to do is really grow our commercial operations again. So invest in marketing and sales, business development, right? To make sure that we're out there and people know who we are. I think that's really what's been the biggest issue for Pluristyx. When we look at, we've even had a third-party analysis done, competitive landscaping. We're right up there with the top players in the industry in terms of capabilities and tools and technology. Where we've been lacking is in that visibility, market visibility, right? So I think a lot of times it's been, who is Pluristyx? And the goal in 2026 is to not hear that question anymore and to become a household name in the industry, if you will. And I think we're already well on the path. Just in the last 3-4 months, we've definitely seen a shift, right? And people recognizing the name, the brand, and knowing who we are and what we do, which has been terrific. So a lot of it goes towards those commercial efforts. But we also continue to, like I said, come out with new products. So it's going also into new product development, as well as expanding our GMP lines. So we have a couple of GMP lines that are ready to be taken into clinical manufacturing. We're looking at expanding that portfolio as well to have more GMP lines. And we recently announced a strategic partnership with Breakthrough T1D, formerly Juvenile Diabetes Research Foundation, where we are now taking that IACT immune cloaking and fail-safe technology and engineering and editing it into one of our GMP lines that is already making beautiful islets and producing insulin. But now that same line will have the IACT stealth and fail-safe technology incorporated, so it's ready for clinical prime time.
Sounds like a lot on your plate.
Yeah, it's a lot of fun.
Which is good. Okay, so for those who want to continue to follow your work, where will you be in 2026?
Yeah, so we will be at JP Morgan. We will be at Advanced Therapies Week in San Diego in February. We will be at the ISCT annual meeting in Dublin in May, at ISSCR in Montreal in June, and then definitely a meeting on the Mesa in October again in Phoenix. There's a high likelihood there are going to be a number of other conferences, probably Biotech Week Boston and a few others that we will be attending as well. So plenty of opportunity to find us. You can follow us on LinkedIn. That usually tells you where we'll be and who we'll be talking to. That's a great place to continue to follow us. Our website, although it needs a refresh, so there will be a refresh coming in 2026, but that'll give you an idea as well. And we are looking at multiple opportunities. We're currently traveling with a number of partners, to do lunch and learns, customer site visits, partner visits, hands-on training. So if any of that is of interest, definitely reach out to me and we can set any of those things up. And we're just looking to, just share our technology, our tools with the industry. And we're all here for one primary reason, right? We're passionate about improving the quality of life and saving lives. And so however we can partner to do that, we are always open to those conversations.
We'd like to thank our guest, Priya Baraniak, Chief Commercial and Development Officer at Pluristyx, for joining us on the Biotech Stack. For more information on Priya or Pluristyx, please check the link in our show notes below. Thanks for listening, and we'll see you next time.